Okay, so one, I want to get a feel for how did this mini lecture go. Now, I asked this because this is always a tough subject, so I want to make sure that we pace this appropriately. Because especially with this concept, and maybe I should have thought this way about molecular bio too, but I want to make sure that there are some very general themes, and especially for those of you in nursing or going into nursing where this is the only taste of immunology you will get right now, there's some really basic concepts that are really important to cover. So I want to make sure that we pace appropriately rather than just speed through this and have you misunderstand everything. So hands raised if you walked away crystal clear with a great understanding of B cells and T cells and activation. I'll wait 2 seconds to see if any hands go on. All right. Hands raised if you have an inkling of what's going on. Hands raised if you don't know what the heck just happened. There's a couple of hands. All right, perfect. All right. We're going to take the time with this. I actually think what I want to do today then is to take that mini lecture and let's expand it out. I want to walk through the individual steps of what's going on here. With that said, we may not get through everything. We may have to move some of this to Friday. So already what I'm planning on doing, if this is okay with everybody, where we have built in 2 lectures for epidemiology, I'd like to just immediately replace those with 2 more days of immunology. Because one of the things that I want to make sure we do have the time to do as well is not necessarily learn more, but go more into depth with how do microorganisms overcome some of these immune responses, okay? And so we'll take some time with that. What's that mean in terms of Friday? For Friday, just don't do the homework. Don't do the mini lecture. Review today, okay? So no homework for Friday. We'll just save that until after break, okay? Any questions on that? Okay, that's 1. 2, I think we need to have an honest conversation about the exam. I have not graded all of them yet. Okay, so this is not a mark on anybody's individual exam or the class's exam as a whole. But let me tell you my personal thoughts when I walked into this exam. I thought, Zach, this is a pretty hard exam. And I think my suspicions were correct, that this was a pretty hard exam. So here's my question, and this is where I want honest and open feedback, and because I don't know the answer to this. How many of you felt that it was fair given what we had done in class? And if you don't feel it's fair, that's fine. But how many felt it was fair? I see some wave hand. Who felt it was completely unfair? Somewhere in between. Okay. All right. And that's what I was afraid of. So when I wrote this exam, I knew that it was going to be challenging because in part I was going back a lot to some of that molecular stuff that we had talked about, and my fear was nobody's looked at this in 2 weeks. And I think those suspicions were correct. So to rectify that issue, here's what I am planning on doing. Just to give you a heads up, my goal is to have these exams back in your hands by Friday. The score you see will be the lowest possible score that you have on that exam. What I'll be doing is I'll be giving you guys over the weekend to make corrections to the exam, and then to hand it back for partial credit. Okay. I have not decided on the due date for that. It will probably end up being Tuesday or Wednesday. I'd rather it be Tuesday because I assume a lot of people are not going to be here on Wednesday or might just show up for something. But since we don't have anything scheduled, I'm not assuming anybody's going to be here. So I will let you know of that day. It'll be a hard deadline for that to hand it back in for extra points on the exam. And at that point, it's completely open book. So to me, it's more important you learn the information than to just toss this exam out and be disgruntled and angry about it. and hate molecular biology, and more importantly, phagocytosis. Because I think that's an important part of understanding what actually happens next. We said phagocytosis helps to, in some ways, tie the innate together with the adaptive immune response. So what are the major steps of phagocytosis? What's first? Attachment. Between what? What are the terms that we use? Between the pathogen-associated molecular pattern, or PAMP, and before lysosome. What's that? The PRR. I'm blanking on the term again. Something receptor. Pathogen. What's that? Crap. Anyway, PRR. Okay. TLR would be toll-like receptor. That one apparently I can remember without a problem. So we get attachment, then we get what? Phagosome forms, phagosome matures, phagosome fuses with lysosome, and then what happens in the phagolysosome? Pathogen is killed, ideally, right? We looked at an example, Mycobacterium, where that actually doesn't happen. What happens after that is where we connect adaptive and innate immunity. So in the case of a neutrophil, after this process of the destruction, that debris just gets removed. It gets exocytosed from the cell. In the case of macrophages and B cells, two cell types that we call professional antigen presenting cells, or PAPCs, they will end up taking those pieces of pathogen and presenting them back on the surface of the cell. Okay? On specialized receptors known as MHCs, or major histocompatibility complexes. There are two major classes of MHCs, 1 and 2. And what we find is that there's a pretty good distinction between the location where the pathogen is coming from and which MHC that the pieces of that pathogen are going to be presented on. You may have heard in the mini lecture I used the term epitope. I may have used the word antigen. I can't remember if I used that one as well. But let's just define those two terms real quickly. An antigen is something that is antibody generating. So that's where the name comes from. An antigen really is in most cases we think of it as a protein from a foreign substance. So it could be it could be a spike protein from a virus. It could be a flagellar protein from a bacterium that our body recognizes as being foreign. An epitope is a subpiece of the antigen. In other words, if you have a protein that is 200 amino acids large, it is an antigen. Epitopes are at about the length of 10 to 20 amino acids. So they only represent a small piece of that antigen. So one antigen can have numerous epitopes. Okay? And each one can give rise to a different set of antibodies. Each can give rise to a different set of T cells and B cells being activated. What is the main distinction between MHC1 and MHC2? And the simple answer is location. But for MHC2, what types of antigens are they presenting? Where are those antigens coming from? Before then. So still think before T helper. Now T helper will interact with MHC2, so we'll come back to that. We'll come back to that. But before we even get that T cell response, we actually have to have these MHC complexes loaded in some way. Loaded with that antigen, or loaded with the epitope. So there are two ways in which this could occur. the one way, no, I don't have this image anymore. Okay. So one way is if an organism, oh, bother. So if an organism phagocytizes, or if a macrophage phagocytizes a pathogen, destroys that pathogen, breaks it into small little pieces, those pieces will bind to MHC2. In other words, MHC2 will bind to antigens that have been broken down by or within the phagolysosome. Okay. What we find then is that once that loading has occurred, we find that this phagolysosome actually kind of gets shuttled to different locations. So one of the events that might happen is we get a exocytic vesicle with MHC2, pathogen or piece of pathogen is loaded into it. This exports to the cell surface so that ultimately what we get is MHC2 on the surface here, piece of pathogen or antigen loaded in that MHC2 protein. And this is the presentation event. And for each pathogen, there's hundreds of antigens, hundreds of epitopes. And so you can imagine that from one bacterial cell, you're going to get numerous MHC, I'll just draw them as shapes here, MHC2 complexes, each with a different epitope being presented. And each of those epitopes has the ability to activate a different T cell. So MHC2 pathogens that are brought in, broken down in the phagolysosome. Oftentimes we find that MHC2 will present bacterial and viral components. Okay, primarily bacterial and viral components. Yeah. MHC1, on the other hand, oftentimes we find disproportionately that it's going to present viral antigens. And that comes from, if we think about the life cycle of bacterial cells and viruses, bacterial cells can replicate on their own. Viruses cannot. What does a virus have to do in order to replicate? Attach to a host cell, penetrate into that host cell, and then we can get biosynthesis to occur. In other words, a virus is wholly dependent on getting into a host cell in order for its replication to occur. And what we find is that during that process of viral replication, the cell is not, all cells in this case, not just professional antigen presenting cells, but every single cell that has a nucleus is going to constantly be flavoring or tasting what's being made inside of it. Some of those proteins are its own. Some of those proteins may be foreign. So let's take a look at the example here in case of a foreign material. So we've got virus inside of a cell, really huge virus apparently. And within the cell, there is a protein complex known as the proteasome. And if you ever look at pictures of it, it's oftentimes drawn kind of like a cylinder that looks like a garbage can. It's actually what its three-dimensional structure looks like, is a garbage can. Organism gets fed in one end, and out the other end comes out these little pieces. Now, we're not going to get into the specifics of how these pieces then get loaded onto MHC1, but eventually what we will find is that these samplings, or these short peptides, Claire, sorry.
Does the MCH2 not have that garbage can thing?
No, so all cells will have this garbage can thing, which the name is the proteasome, but MHC2 is only loading pieces of antigens from within phagolysosomes. All right, so MHC2 loads from phagolysosomes. MHC1 is actually loading things that are found in the cytoplasm. So there's kind of there's a spatial differentiation here on the products that they're loading. So these products get broken down. Eventually what we will find is that they get presented on MHC1. Another way to define this is that MHC2 oftentimes will present pieces of antigens that come from extracellular components. And that should make some sense. They're being brought from the outside, phagocytized, broken down in a phagolysosome. Whereas MHC1 is presenting antigens that are internal to the cell. So presenting proteins that are being actually physically made within that infected cell. Devin.
Is that kind of along with like, I forget what it was, but there was something where it's like, once like the pathogen or something is in it, like the antibody can't have an effect on it or something?
It's a really interesting point. Let's come back. Can we table that for right now? That really becomes important when we talk about T cells, T cytotoxic, the T helper, and the B cell response. Absolutely. Okay. So far we've talked about major difference between MHC2 and MHC1. MHC2 loads external pathogens from phagolysosomes. MHC1 internalized pathogens from the cytoplasm. Okay. The next question becomes, and let's just revisit too, what cells are these actually found on? Now, in every single cell you have the genetic information to produce MHC1 and MHC2. However, what we find in terms of the cells that actually express these individual genes, MHC1 is found on every single cell, with the exception of red blood cells. And MHC2 is only found on those professional antigen-presenting cells. In the case of what we're talking about in here, that includes B cells, it includes macrophages. It does not include neutrophils. Okay, any questions with that? T cells are just recognizing. Good question. So T cells just recognize MHC2. T cells, if I'm not mistaken, should express MHC1 since we typically categorize all nucleated cells have an MHC1. All right. The next question becomes, sorry, Meg.
Did you say that the professional presenting or
Presenting. So the next question becomes on which cells do interact with MHC2 and MHC1. For this, maybe the best way to do this would be to draw a table and then come back to that figure. So what we find is that the cells that are activated or that become stimulated by recognizing a specific antigen loaded into MHC1 are our cytotoxic T cells, or our TC cells. There are a couple of important role players here, and I'll try to draw this out as well. The T cell has two receptors that are important to this process. One of those is known as the T cell receptor, or sometimes it's abbreviated TCR. And the second receptor that is important here is known as CD8. You do not need to know what CD means. but we oftentimes call CD8 a co-receptor. So what do they recognize? Together, they recognize MHC1 and the specific antigen localized in it. Now, one of the things that we said about the innate immune response is it recognizes patterns. It recognizes teichoic acid. It recognizes lipopolysaccharide, things that are common among different pathogens. The adaptive immune response, on the other hand, T cells, B cells, recognize very specific features. The way that they identify these specific features is encoded within our genome. It is way too complex for us to take the time to understand right now. But what you should know is that each T cell and each B cell that is produced is designed to recognize a different specific antigen. And the T cell, and what we get here is an interaction between the T cell receptor and that particular antigen that's within MHC1. It's kind of like the same way that we talked about a substrate has to fit into the pocket of, or fit into the active site of an enzyme, or an antibiotic can compete for the binding to that enzyme. It's those types of interactions, kind of like a hand and a glove, that are important here for the interaction between the T cell receptor and this specific antigen. Okay. CD8, on the other hand, recognizes actually the generalized structure of MHC1. In other words, for this T cell, this cytotoxic T cell to become activated, you have to have two signals occurring here. One is that T cell has to recognize the antigen. The second one is CD8 has to recognize MHC1. And if both of these are recognized, then that's going to activate the T cell and now allow for it to perform its function. And we'll come back to that function later on. Allison. Yeah, so one is, so again, two things have to happen at the same time. The T cell receptor has to bind to the specific antigen, and CD8 has to bind to MHC1. And the only way that this occurs is if that specific antigen is loaded within MHC1. When this happens, then, what we find are a couple of things. That T cell becomes activated. One of the ways that it does is we find these cells secreting cytokines that activate those T cells. Then they disconnect from each other, and now that T cell can start to perform its function. And we'll come back to that later. Devin.
The cell with the TCR receptor?
Yes.
Like, then
So it's more that those cytokines are going to take this T cell from it kind of like a resting state to now an active state. So main terms to associate with MHC1, CD8, T cytotoxic, T cell receptor, internal pathogens. Okay. Questions on that side? We'll take a look at MHC2 in a minute, but I want to just see if there are any other questions here. Okay. May I, maybe I've got enough room to do this. We'll just mirror it. MHC2 interacts with T helpers, or T sub H. The role of T helpers is just as the name describes. They're going to help, really what they're kind of doing in some ways. You can kind of think of them as like a boss in a business where the boss doesn't do the work, the boss tells everybody else what to do, and that's what a T helper cell is doing. It's basically orchestrating or telling other cells in the immune system what to do. And we'll come back to that again a little bit later. But their activation is somewhat similar to the activation of MHC1. On MHC2, the epitope might be completely different than the one found on MHC1. Again, we have a T cell receptor. And again, we need a co-stimulatory interaction, or a second interaction between the T helper cell and the MHC2. And that interaction is with a molecule known as CD4. You may or may not, probably may not, remember we actually brought up CD4 really briefly at some point. CD4 not only is a co-receptor for this interaction, but also is what HIV binds to. Okay, and so T helper cells are the tropism, or are the cell hosts for HIV. Only cells in the body that express this particular molecule. So same thing happens here. We get an interaction between MHC2, CD4 with MHC2, the T cell receptor with the particular antigen. Macrophage secretes signals that's going to activate that T helper. So it secretes cytokines. And actually in return, the T helper does the same thing right back. It secretes cytokines that enhances the phagocytic activity of that macrophage. So here is where we start to see the first feedback between the adaptive and innate immune response. T helper becomes active, feeds it right back, activates that macrophage to increase its power to destroy microorganisms. Okay? Questions on that? Okay. I'll just pause for a minute so that if anybody wants to finish taking notes or drawing anything, they can, and then I'll move ahead. All right, so the next question becomes, what do T helper cells and what do TC cells or cytotoxic T cells do? Let's start with this, because this actually was kind of part of the homework and having you describe structure and function of them. What I'd like you guys to do is just turn to somebody for 2 minutes, describe what is the function of a T helper cell, what is the function of a cytotoxic T cell. Yeah, really what it means, there are 2 subtypes. Actually, there's even more. There's 4 subtypes of T helper cells that are all defined by like what they respond to and the receptors on the surface. All this image is trying to say is, like, in general, T helpers all do the same thing. They will enhance or activate the immune response. but there are some designated as the T helper 2 that activate B cells to produce antibody. There are some designated as T helper 1 that increase what we would call a cell-mediated response, and that cell-mediated response is phagocytosis. It's activation of cytotoxic T cells. So, I mean, it's a distinction that I'm not really sure if I don't think I'm concerned with you guys knowing. Yeah, and we'll talk about that role briefly, but its most important thing is to know that they activate those other cells. Oh, gosh. No, did I say they did? Because I think they do. Yeah, it is up there. Yes, they do. But the mechanism by which it occurs, I can't remember right now. It might just be to secrete cytokines that kind of put the NK cells on notice.
Do the NK cells identify the harmful cells?
They do. We'll come back to that. We might cover it today. We might have to wait until Friday, but we'll cover it. All right. So, what is the major role of a cytotoxic T cell? It kills an infected cell. I mean, bottom line, that's exactly what it does. So we'll talk a little bit about how that occurs. But again, remember what the cytotoxic T cell recognizes. It recognizes foreign substance or foreign antigen loaded in MHC1. So, if a cell is presenting something that is considered foreign, the cytotoxic T cell will bind to that MHC1, CD8 binds this, and these would be T cells that are now already activated. They've gone through the process of being activated. And what the CD8 cell, what this T cell does in return is it just starts to produce these exocytic vesicles, already has them kind of preloaded, and just releases everything from them. And there are two chemicals, granzymes and perforin. Perforin pokes holes. It perforates a cell. Basically, it disrupts the plasma membrane of that cell. The granzymes trigger a process in a cell known as apoptosis, or sometimes it's referred to as apoptosis, which is, in this case, the absolutely wrong way to say it, if you look back at the Latin or Greek, okay? What is apoptosis or apoptosis? Programmed cell death. In other words, what's happening here is this cytotoxic T cell tells this infected cell, time to die. What's the benefit of that? It stops growing, which means what happens to whatever inside of the cell? If it's a virus, it dies as well. It loses its host to replicate. In other words, this is kind of like, let's kill one cell to save the host. That's kind of the design behind this. All right? By killing this one infected cell, it blocks the replication of the virus from proceeding any further, and therefore prevents the spread or production of new viral particles that can go and infect more cells. That's the design behind this. Perforin just pokes holes in the membrane of the infected cell. And so this is kind of unidirectional. What we find is that all of these, and this interaction is incredibly close, so there's very little collateral damage. These perforins and granzymes are basically being directed into this infected cell. The poking holes actually helps to contribute to that programmed cell death. And there's thought to be more than just that as well, but that's kind of being uncovered right now. Okay, so that's the function of CTLs. T helper function. Basically, yes, it activates the immune response. So T helper cells, there are right now, as far as we know, 4 different subsets of T helpers. known as T helper 1, T helper 2, T helper 17, and I think the last one is 4 or something like that. The nomenclature is horrible. And to be completely honest, we don't know what all of them necessarily do. The two that folks most often concentrate on are these T helper 1 and T helper 2s. I am not concerned with you guys necessarily knowing which one is which, okay? But I'm just pointing this out as there are subsets of T helpers. The major role of the T helper is once it has been activated by a macrophage, once it has received cytokines from a macrophage telling it to start to create this immune response, the T helper cell will begin to activate CTLs, macrophages, neutrophils, NK cells. And the way that it does this is T helper cells start to secrete cytokines. Whenever they bind to a macrophage, from this point on, whenever they bind to a macrophage or a professional antigen-presenting cell that has that same exact antigen on it, loaded in MHC2, what that T helper cell is going to do is just start secreting cytokines left and right. And in doing so, those cytokines activate or turn on or enhance the activity of the phagocytes, the neutrophils and macrophages. Increase the killing capacity of cytotoxic T cells and NK cells as well. In the case when a T helper cell binds to a B cell and binds to MHC2 on a B cell, well, it takes this B cell from a naive state and now activates it as well. And we'll talk about that activation of the B cell probably on Friday. Okay. So T helpers, they regulate this response. It's like that really annoying guy at the party, or girl, let's not be specific here, who is trying to give everybody drinks. Drink for you, or Oprah Winfrey. Car for you, okay? It's that kind of person who's trying to get everybody involved. There are times in which viruses are amazing because they have ways to get around immune detection. One of the common ways is to actually prevent MHC1 from ever making it to the surface of a cell. So there are a couple of classical examples of this. One of them is human cytomegalovirus, which is related to herpes and EBV and chickenpox or varicella zoster virus, etc. And what this virus does is, instead of allowing for MHC1 to be loaded with its antigens, it actually prevents this process of that MHC1 ever being brought to the cell surface. And so these cells are void of MHC1. What effect does that have on the immune response? Particularly, what effect does this have on cytotoxic T cells or detection of this viral infection? What's that?
Does it not release the cytokines?
True. Think back to the role of cytotoxic T cells. What do? They kill. If MHC1's not there, will this cell die from a CTL? No. Because a CTL needs what? Well, it needs that MHC1, it needs that antigen loaded into MHC1 in order for this process to occur. And so in cases where viruses are preventing MHC1 from being properly displayed on the cell surface, CTL can't kill it. How does the immune system make up for the lack of MHC1 on the surface? There's one cell type we haven't talked about yet. And it's not B cells. What's that? It's the NK. What's NK stand for? Natural killer. All right. The natural killer cells recognize this absence. So here's an image of normally the interaction between an NK cell and a normal cell. This normal cell has MHC1 presenting some antigen on the surface, might be self, might be non-self. And the NK cell has two different types of receptors. It has an activating set of receptors. It has an inhibitory set of receptors. When the inhibitory receptor recognizes or binds to MHC1 and the antigen, that silences or keeps quiet that NK cell. For any of you planning to have children, pacifiers are your best friend. A pacifier is like the inhibitory signal to an NK cell. But when you remove that pacifier, what happens? The baby goes crazy, and that's what an NK cell does. An NK cell, when that inhibitory receptor no longer binds to MHC1, activation. All fury breaks loose. And what we get is this NK cell now actually performing the same exact function that a cytotoxic T cell does. Secretes perforin, secretes granzymes, and the secretion of those two kills a cell that does not have MHC1 on its surface. And so our immune system has built into it a mechanism to not only recognize a foreign substance on MHC1, but also has a mechanism to recognize the complete absence of MHC1 from a cell's surface. This is important not only in terms of infection, but if you're these days where we're seeing a big movement in immunology is actually in cancer therapy. And there's a lot of heavy research being done right now on cytotoxic T cells and on NK cells and their potential role in killing or treating tumors that are presenting mutated forms of a protein that might be recognized as foreign or are presenting no MHC1 at all. We do see both of those happening in some cancers. Okay? So there's a lot of exploration with this. Okay, so let's just real quick, let's take 1 minute. Compare and contrast NK cells and cytotoxic T cells, not only in structure but in function. Okay, take about a minute to do that. All right. I'm not going to ask for responses. Keep them to yourself for now. Maybe we'll take that to review on Friday. But let's just kind of cover the major things we've talked about today. And I want to go back to the objective slide on this one. Just to, again, keep in mind what we're talking about here. We did actually talk about the influence of a virulence factor on pathogenicity. We didn't name it, but we said there are some viruses that prevent MHC1 from being presented. And we talked about the effect of that then on how the immune system would respond with either CTLs or NK cells. We began to talk about the function of at least a couple of these here, T helper and TC. We'll talk about B cells on Friday, so let's make that our primary focus. I just had you doing a little comparison between TC and NK cells. And then 11.6, how antibodies enhance that immune response, we'll cover on Friday. So a couple of things just to kind of remind you, nothing due for Friday. Last question, anybody in here not like Greenbush? I was thinking Monday, to entice you to actually come and work for a while on those presentations, I'll buy a variety of Greenbush. We all cool with that? We need a little pick-me-up in here, so we'll do that. All right, have a great day. I'll see you guys on Friday, or tomorrow or Friday, one or the other.